Welcome to the summary video for the HIV pathogenesis lecture. This lecture is got a lot of information in it, multiple parts to it. I really like the videos for this particular lesson, but there are a few points that I want to kind of make sure that you get and try to fill in some of the spots that the videos might gloss over a little bit quickly. So first of all, I want to emphasize for this lesson, as well as really all of the other lessons in this class, we're learning about this material in a contextual sense, and that today we're going to focus a majority of our efforts into the adaptive immune response, but we're talking about the adaptive immune response in the context of HIV and directly applying what's happening in a person who has an HIV infection, what's directly happening to their adaptive immune response that results in the clinical presentation and the complications that we see. With that in mind, you are not expected to be HIV experts after this. You don't need to know the gory details of all of the minutia that we know about HIV. And that's true of every one of these diseases, just like you already saw with influenza and you will see with other things in the coming lessons. So with that in mind, the overview, the HIV pathogenesis overview video, I think he does an exceptionally good job. However, that particular video really emphasizes the CCR5 and CXCR4 co-receptors, and that gets into a level of detail that you just really don't need to know for this lesson. So don't get lost in, those nitty-gritty and trying to memorize all of that stuff. Catch the big picture of what HIV is doing in the cell and how all of that results in the complications for the rest of the body and the clinical presentation. So HIV human immunodeficiency virus does a really cool thing that most animal viruses don't do, and that is that it can integrate its nucleic acid into the cell's DNA. Remember, we saw this with phage and the lysogenic cycle, but animal viruses don't do that very often. When the viral DNA is inserted in, it's referred to as a provirus. It integrates into the host DNA. And HIV is particularly interesting because it's actually an RNA virus, but it wants to insert itself into the host's DNA. So it takes its RNA that it comes in with and it converts it into DNA, which is weird. Most of the time, DNA is converted into RNA, which is then read to produce a protein. So this is backwards of the normal process that happens in cells. So in order to accomplish this unique step, HIV has to carry a special enzyme. So that enzyme is called reverse transcriptase, and it's only found in a very small subset of viruses and organisms in the world, because it's not something that's done in the typical flow of a cell. Okay, so like I said, the first video I think does a good job of doing the HIV pathogenesis overview, basically kind of how it walks through the cell and then how it moves its way through the body. It comes in, it can attack and infect a small staple of immune system cells, which then carry it to the lymphatics, where it can infect even more immune system cells. And this has a devastating effect for a couple of reasons. First of all, remember that when a virus enters into a cell, it takes over that cell, and then when it leaves the cell, it typically kills the cell that it's taken over. Well, so if it infects an immune system cell, then it generally kills that immune system cell as it's leaving, which means a dead immune system cell. The other thing to remember here is, and what will be emphasized in this lesson, is that when you have one of your body cells that are infected by something, that infection is shown on the outside of the body cell. And that serves to target the immune system to that infected cell to kill it. So here the virus is killing some of your immune system cells when it leaves the cell, when it buds off the cell. And it's also targeting other immune system cells to end up killing it because it's infected. So you end up with a reduction of these key immune system cells, which then reduces the efficacy of the immune system as a whole and leaves the person very susceptible to infections that would otherwise be fairly easily contained and fairly easily fought. Okay, so You get the overview of the pathogenesis, the way that the virus moves around the body, and how that results in some of the devastating clinical effects that are seen. In order to understand why we see that reduction in the immune ability and the immune activity, you have to really understand the function of a key group of cells, one in particular called the CD4 helper cell. And I do want to point out here that there's, depending on the source, the video or the textbook you're reading, sometimes these cells are called helper cells, sometimes they're called CD4 cells, same thing. CD4 cells are also known as helper cells. And just to help clarify on that note, there's another type of T cell that you're going to learn about in this lesson, which is called a cytotoxic T cell, and it's also sometimes called the CD8 T cell. So both CD4 and CD8 cells are helper cells, but are T cells, but they have different functions. The CD4 cell is a helper T cell. The CD8 cell is a cytotoxic T cell. We'll see that the helper T cell is absolutely vital in memory production in the specific immune response for both types of specific immune response. CD4 helper T cells are kind of the heart of the ability of the adaptive specific immune response to create memory. On the other hand, CD8 cytotoxic T cells are cells that will kill infected host cells. So our body cells that have become infected or affected will be killed by these CD8 cells, very much like we saw with the innate immune system and NK cells, natural killer cells. So for the videos in this lesson, there are a couple of things that I would like to point out. First of all, in this lesson, I've got reviews of the innate part of the immune system in there. It doesn't ever hurt to hear it lots of times. They all overlap. We're going to talk this material over and over and over throughout this semester. So take 10 minutes to review the first half of the immune system, the innate part. And then I've got several different videos for the adaptive specific part of the immune system too. These videos are redundant in that there's a Khan Academy video and then there are a couple of Crash Course Biology videos. I recommend you watch all of them. It does not hurt to hear it multiple times. In fact, this type of material, these videos, you may need to watch over and over and over to let it kind of sink in, because it's detailed material that you've really got to get a good handle on. I do want to point out, though, sometimes I love the Crash Course Biology videos, especially these. The immunology ones are really, really great. But sometimes people complain that he talks too fast. So I wanted to give you a little tip in case you haven't discovered this. When you go to the tool icon on YouTube, there's a playback speed adjustment. You can make it go slower or you can make it go faster on any of these videos. So if he talks too fast for you, besides just pausing it over and over and over and rewinding it, you can actually slow down the talking speed so that you have plenty of time to take copious notes on everything. Okay, so coming back to what we were talking about with the adaptive part of the immune response. So what happens here is you get a foreign body, and bacteria, virus, fungus, whatever, comes into your body, and it's got certain features on the outside of it that we refer to as antigens. Your phagocytes, your macrophage, and also your dendritic cells. Macrophage and dendritic cells can work interchangeably in this particular job. They will eat the foreign body, grab one of the antigens, and present it on the outside of the cell. So basically they eat it and then show everybody what they just ate. That macrophage or dendritic cell will then activate a helper T cell. So helper T cells float around in the area. They are drawn through inflammation. They go to the site of the phagocytosis of the eating. Helper T cells have a T cell receptor on the outside of them that matches a specific antigen. So if the helper T cell receptor matches the antigen that is on the outside of the foreign object, then that will activate that helper T cell. and it will start releasing lots and lots of chemicals. I do want to pause for a moment and point out that there are lots of names for these chemicals, okay? This is immunology. It's got a whole vocabulary of its own. In this particular picture, you see the term cytokine. Cytokine, chemokine, interleukin, just a few of the common ones that you'll see. All of these terms mean very detailed things for immunologists, but for our class, you can think of them as just chemical signals, okay? So our T helper cell, whose receptor matched the antigen on the outside of the foreign thing that was being presented by the macrophage or dendritic cell, will start releasing these chemical signals. Those chemical signals do a couple of things. They activate other cells involved in the adaptive immune response as their primary function that we're going to focus on in this class. So let's first begin with the humoral, the antibody response. So these chemical signals coming off of the T helper cells can interact with B cells in the area. But let's first talk about what these B cells are and why they're there. So remember that B cells, you have in the early parts of your life, each B cell will pick a particular antibody that it will make, and that antibody will match a particular antigen. So for all of the different antigens that you may come into contact with your life, hopefully you have a B cell that can grab on to that antigen. And the very, very first time you see an antigen, you only have one B cell to match it, most likely. And so what happens is your immune system will be drawn to the site through inflammation, will be drawn to the site of that phagocytosis, drawn to the site of that infection. And the B cells that are in the blood and the B cells that are in the area will be drawn over there, and they will check to see if their antibodies can bind on to the antigens on the outside of the foreign thing. Most of them will not be able to. But eventually, usually 3 to 5 days later, you will find one that can match. Their antibody matches the antigen on the foreign object. And when that happens, that B cell starts releasing chemicals. So you've got your T cell that matched the antigen being presented by the macrophage or dendritic cell. You've got your B cell that matched the antigen on the outside of the foreign thing, and both of them are sitting there releasing chemical signals. Well, those chemical signals attract the two cells together. And the two cells see if they match, if what the T helper cell responded to is the same thing as what the B cell responded to. And if it is, then that sets off a really interesting process called B cell expansion, where basically the B cell copies itself lots and lots and lots and lots of times. And the B cells that are produced have the same antibody that was on the original B cell that matched the infection. So these B cells that are produced are all going to have that same antibody. Of the B cells that are produced, they are not the exact same type of B cell as the starting kind of parent cell that produced them. This first cell up here, this first B cell, the one that was drawn to the site, is referred to as a naive or virgin B cell because it's never been activated before. It's never seen its antigen. The babies that cell makes through B cell expansion are no longer virgin B cells anymore. They're slightly different versions of that B cell. So there's really two populations of B cells that will come off of that naive B cell when it expands. There's a group of cells called plasma B cells, and there's a group of cells called memory B cells. Okay, so the plasma B cells versus the memory B cells, they all have that same antibody that was originally found on the naive B cell, except they have slightly different jobs. The plasma B cells are antibody production factories. They produce several thousand antibody proteins a second. And which antibody proteins do they produce? That one, the one that matched the antigen on the outside of the original infection. These plasma cells only live a few weeks. So they produce huge amounts of huge amounts of antibodies, and then they die off. What's left behind a few weeks later are these memory B cells. These memory B cells also have that same antibody on the outside of them as the original one that matched the infection. Okay. But what makes them different than the original naive ones is these guys don't require T helper cells anymore to go through B cell expansion. So if these memory B cells come directly into contact with a foreign antigen, that will set them into B cell expansion and produce more plasma and more memory. So it takes significantly less time because you skip the T helper cell step entirely. So the memory B cell comes directly into contact with the infection. Plasma cells are produced to make antibodies, and more memory is made. So you have even more copies of that memory B cell floating around for the next time that that organism tries to come in. Okay, so that is an overview of what happens the very first time that you see an antigen for B cell production, antibody production, and the humoral immune response. And you can see the T helper cell is absolutely vital in getting that process started and producing the B cell memory. T helper cells are also vital in the cell-mediated immune response, the intracellular killing cells that are infected. So quick overview of that. You have one of your body cells, maybe it's a muscle cell or a liver cell, whatever. It gets infected by a virus. That cell could be killed by an NK cell that's looking for abnormalities in the proteins that are expressed on the outside of the cell, like no MHC1 or interferon proteins that are produced on the outside of that cell. But also, what that cell will do is on its MHC2, it will actually present a piece of the virus that infected it. So here you've got, a normal muscle cell or whatever that's got a piece of virus sticking off of it. And that piece of virus triggers the immune system to come and look at this cell and see what's happening. So the cells that are particularly scanning the MHCs looking for pieces of viruses are these cytotoxic T cells. And the reason they're looking for particular pieces of viruses is because their T cell receptors match a particular antigen, just like we saw with the T helper cells. So with each infection, there are certain antigens on the outside of it that will be presented when the cell gets infected. And one cytotoxic T cell, also known as a CD8 T cell, will match that particular antigen being presented. And when that T cell receptor on the cytotoxic T cell matches the virus being presented by that cell, it triggers that cytotoxic T cell to produce perforin and granzyme, which kill that cell. So again, this process is basically exactly the same thing as what natural killer cells do, except what triggers the activation. So for natural killer cells, you're looking for interferon proteins on the outside of the cell. For cytotoxic T cells, you're looking for a specific antigen being presented on the outside of the cell. The T helper cell comes into play with the cytotoxic T cell in a couple of ways. First of all, when that T helper cell gets activated and excited, it further activates the cytotoxic T cell to make it a super good killer. But it also, those chemicals being released by the T helper cell allow that cytotoxic T cell to produce memory. So unlike NK cells, which kill a cell and move on, cytotoxic T cells, when they kill a cell, they will then copy themselves. So then there's multiple versions of that cytotoxic T cell around. Except these memory versions of the cytotoxic T cell are now fully activated super killers, even without the T helper. Like the original one had to have that T helper help. Memories don't have to have that. The next time that that antigen shows up on a cell, the memory cytotoxic T cells will kill that cell and reproduce themselves. So you end up with an army of them all going after that one particular antigen, that one particular virus or whatever it is that's inside of those cells infecting them. Okay, so that is kind of the overview of activation of the specific immune response, the adaptive immune response. There are a couple of other little things I want to emphasize before we kind of give up on the summary and let you dive into the heart of the lesson videos. The next thing I want to emphasize are the antibody functions. So there are really 5 key antibody functions that we focus on in this class. They are N, I, A, O. N stands for neutralization, which the lesson videos do a good job talking about neutralization and agglutination. Neutralization, we actually saw a little bit with viruses where the antibody can bind onto the virus, which then keeps the virus from being able to bind onto the cell receptor. So then the virus does not match the cell receptor anymore and can't enter into the cell through endocytosis. The same basic theory applies with exotoxins that we'll learn about in future lessons. I stands for immobilization, where the antibody can bind onto the motility structures like flagella or cilia, if you're looking at a eukaryote, and that can keep those motility structures from being able to go, and so then they can't swim away. The first A is for agglutination, which is basically clumping together the foreign things, making a giant mesh of them, which makes it easier for the phagocytes to come and eat. The second A stands for activation of complement and inflammation. And basically the concept of this is these antibodies float around in our bloodstream and should not have anything to stick onto. Their job is to stick onto foreign things that are not supposed to be in our body. So if you have an antibody that's actually attached to something, that serves as a signal that there's something foreign, and the immune system will come rushing over and it will set off the complement system, which will set off inflammation and a whole bunch, a whole cascade of reactions that will happen because the antibody is bound onto something in your body. And then the final function of antibodies, O, is opsonization. This is basically sticking on the outside of the foreign object, and it allows a handle for the phagocytes. So this is particularly significant if you have like a bacteria that has a capsule, a smooth, hard shell on the outside that's hard to grab. So the antibody will stick on to the organism, and then the phagocyte can grab onto the antibody and use it as a way to grab and eat the foreign thing. So these aren't the only functions of antibodies, but these are the key ones that we're going to focus on in this class. And then the final part of antibodies that I want to point out tie back into vaccines and kind of the timing of disease presentation that we see clinically. So remember, we're just talking about the activation of naive B cells and the full activation of naive cytotoxic T cells. Both of those require the activation of a T helper cell to get the process going. Well, that whole process, the finding the one B cell that matches the antigen, or the one cytotoxic T cell that matches the antigen, and finding the one T helper cell that matches the antigen in that particular infection, that takes time. That leaves the adaptive immune response, almost non-functioning for several days when you're first exposed to that antigen the very, very first time. After that 3 to 5 days, even up to 7 days, your body starts producing memory cells along with the antibodies. So you'll see your antibodies go up, but once the plasma cells die off, those memory cells are left behind. So the next time that you see that organism, you have memory cells there that don't require the T helper anymore. And so those memory cells can start producing plasma cells and start responding much, much faster. So within just a couple of days, you start getting really hefty antibody production. And what you see is with repeat exposure, so the first time you see it, it's referred to as primary exposure. With secondary and subsequent exposures, you end up with an even higher level of antibodies being produced than you started with because you have more than one cell. So at first, you only have that one B cell that you have to pluck. Here, you've got a whole variety of copies of that cell in the form of memory cells. And every time you get exposed, you make more copies. So instead of just activating one cell, you can activate a bunch of the different memory B cells, and you get much higher levels of antibodies produced. This is what vaccines do for you. Vaccines give you the antigen in a controlled sense so that you can go through this naive activation in a protected way. And then when you come into contact with the really aggressive, nasty pathogen, you are already at the memory level and don't have this lag of lack of protection that you do at first exposure. The other thing I would like to point out using this chart is some of the terminology you might see clinically. So another name for antibody is immunoglobulin. And you see this really common clinically with treatments and medicine. So if you see, on the IV bag for your patient, you see IgG or you see IgM. Ig is shorthand for immunoglobulin. So what that's saying is that it's an antibody type G, antibody type M that you're giving to your patient. And the different types of the antibodies, if you're curious, feel free to read up with it in the textbook. But that's a layer that we don't really need for our class. Okay, and the final part of this lesson to talk about is what happens after the immune system starts getting depleted due to the virus. So your helper T cells start dropping, your macrophage levels start dropping, everything starts kind of plummeting because the virus is doing its damage. And that leaves your immune system very weakened, and you start seeing opportunistic infections as you get closer and closer to AIDS. So this important set of vocabulary words, opportunistic infection or opportunistic pathogen, as opposed to a frank infection or a frank pathogen. A frank infection is one that can cause infection even when your immune system is at full speed. Opportunistic are infections that you only see when the immune system is weakened for any reason. It doesn't just have to be HIV, but anything that weakens the immune system can create a situation where you can get an opportunistic infection, which otherwise wouldn't be able to cause disease. Our body would shut it down. So we're going to go over a few. The lesson video for the opportunistic infections, I think he does a really, really good job of kind of setting all this up. I recommend that you look back at the PowerPoints for this lesson to help emphasize kind of what parts of each disease I'm expecting you to know. I would expect you to be able to recognize like a case study on these diseases, what they look like in a person, what the etiological agent of them is. So kind of focus on that as you're going through there. But I do want to point out that while we're just focusing on a handful of these kind of common AIDS-associated opportunistic infections, I do want to point out that this is a very, very small snippet of the ones that are seen. Here's a list of ones that are commonly seen. The list actually could be even longer. We're only going to focus on the most common of those, but the list is significantly longer. So the HIV virus is just devastating to your immune system by taking out a few of these key cells, and it just basically cripples your immune system and makes you so susceptible to other infections. So good luck going through this material. Again, be patient with yourself. Watch the lesson videos as many times as you need. Take lots of notes. Keep referring back to the outline. Keep referring back to the PowerPoint to keep yourself oriented and to remind yourself of how much detail you need, how deep you need to go into this material.